Ngā mihi nui and welcome from Radio New Zealand National. Here's Our Changing World.
Millions of people worldwide experience bouts of depression, and the World Health Organization ranks it as the leading cause of disability.
Cognitive neuroscientist Gina Grimshaw and her team at Victoria University investigate how asymmetries in brain function influence the way we process emotions and how that can be used to predict vulnerability to depression.
Veronica Maduna meets the team in their lab, where they put healthy volunteers through a number of tasks to better understand how vulnerability can turn into illness.
This is a lead-lined chamber. It was actually donated to us by the Department of Defense. I'm not exactly sure why they had one.
So it's a completely sealed...
Not hermetically sealed or anything like that, but it is lead-lined. And so it shields from electrical activity from other sources in the environment. So because what we're trying to do is measure very, very tiny electrical signals on the surface of your skull, in fact, there's much more electromagnetic radiation floating around in the air around us. So this minimizes a lot of the sources of that. So we bring our participants in. And
volunteers, I gather?
Yeah, they're volunteers. They're first-year university students. And we put this very fashionable hat on them, so you can see what it's got are a number of electrodes. And what those are doing is measuring electrical activity on the surface of the scalp. So what you see are these waves of electrical activity as someone is thinking that will pass over different spots on the skull, and that's what we're recording. Then while they're sitting there, they're going to participate in a number of tasks, and we're just going to measure their brain activity while they're doing different sorts of activity. The other thing that we do is just have them sit and relax for 4 minutes, and that's where we're collecting some data on their resting brain state. And that's the one that seems to predict vulnerability to depression. So we're using that as our trait measure, and then we're relating that to different aspects of processing when we ask them to process emotional faces or emotional words or emotional pictures. We actually don't study anyone who is depressed in this particular phase of the project. What we're interested in studying is this relationship between the brain asymmetry and how people process emotional information when they're healthy, so we can determine if that pattern plays a role in how they respond to stress in the future. So it's much more about understanding that relationship between patterns of brain activity and emotional processing than understanding depression itself. One of the asymmetries that we're particularly interested in are these asymmetries in function of frontal areas of the brain that seem to be particularly important for how we process emotion. And we've actually known for decades that the frontal cortex is really important for emotional processing and emotional regulation. And we've known even going back to the early part of the 1900s, that people who've had strokes affecting the left frontal area of the brain are more likely to become depressed than people with strokes in the right frontal area of the brain. And we've known probably for about 20 or 30 years now that there are resting differences. So when you're just sort of chilling out doing nothing, there's differences in brain activity that differ between individuals, whether they show more left than right frontal activity or more right than left frontal activity. And we've known for a good period of time that that predicts vulnerability to depression. So people who are depressed are more likely to show this shift towards more right frontal than left frontal activity. And after they're depressed and when they go into remission, they're still showing that same effect. So it's not actually the depression per se, it's more something about their particular brain's pattern of being. And there are a number of studies.
Does it change in any way during an episode?
No, so it seems to be a stable trait. So your brain, your left and right frontal hemispheres also show some effect of emotional state. So if I make you very happy, you'll show an increase in left frontal activity, or angry actually. If I make you very sad or fearful, you show an increase in right frontal activity. But underneath that, everybody has a stable pattern that's relatively stable from time to time, whether they're depressed or not depressed. And the idea that this is really a vulnerability factor comes from other studies showing that you see it even in the infants of depressed mothers. And in those children, it predicts that they're going to develop into shy and inhibited children who we know are at higher risk of depression. So these seem to be very inherent individual traits of brain activity.
Which of course brings me to the question about genetic influences. It is inherited at least partly.
Yeah, so when we talk about vulnerability to depression, we do know that some people are more vulnerable than others. So if you think about what causes depression, it's easy to see that the first episode of depression is going to be preceded by a period of significant life stress. But we all know examples, and the data bears this out, of lots of people who live under lots of stress and don't become depressed. So it can't just be the stress that's the factor. So some people are more vulnerable than others, and that's our sort of starting point for trying to figure out, what does that vulnerability mean? How is it related to the brain? How is it related to thought? How is it related to emotion? So when we talk about what makes some people more vulnerable than others, we can answer that question on all sorts of different levels. And one of them is clearly a genetic component. So the gene that certainly gets the most airplay right now is the serotonin transporter gene, but it's sort of one of a family of many genes that definitely plays a role in the setting up the serotonin system, which plays an important role in emotional processing and emotion regulation. And there are two alleles, an S allele and an L allele. And so people who have one or two copies of the S allele seem to be more likely to develop depression, but only in response to significant life stress.
So it needs a trigger of some sort.
Yeah, so this is where we see the gene-environment interaction. So if you look at data that's come out of the longitudinal study that's been done in Dunedin, but a number of other studies now around the world corroborate this, what you see is that in individuals who carry this S allele, the more life stress they experience very early in life, the more likely they are to become depressed as adults. Whereas individuals who have two copies of the L allele, in some ways, seem to be very resilient. It doesn't matter what levels of life stress they have, they don't show anything above sort of a baseline level of depression. So it's not the genes that are causing depression, and it's not the environment that's causing the depression. It seems to be the two working together. And that's something that seems to be happening very, very early in development, probably when the brain is setting up these circuits that allow the developing child to deal with this sort of conflicting goals. So if you think about emotional information out there in the environment, it's continually giving us cues about how we should behave or how we should respond. So should we be afraid? Should we be withdrawing? Should we be approaching? Should we trust? Should we like? Should we not like? That's what emotion does for us, is it gives us cues about how we should behave. And so you have all this information coming in, but you also have your own goals and your own needs that can tell you, you know, I can ignore that tempting cookie because it's actually not good for me. Or I can ignore that scary looking person over there because I know they don't really mean to be looking at me. And so we have this top-down ability, and that's really what your frontal cortex is most important for, to control how you're responding to these environmental cues.
So in somebody during an episode of depression, is it this top level that somehow stops working?
It likely is an interaction between the bottom level and the top level. So cognitive theories of depression, which are probably the most predominant, sort of dominant theories in the field right now, suggest that people who are vulnerable, likely as a result of gene-environment interactions early in development, setting up these circuits that allow this negotiation between environmental drives and top-down goals. So that's what you need. To be able to control your behavior and to control your emotions, that if that system is not set up and functioning very well, you can have a system that can get hijacked by the incoming information. And particularly people who are vulnerable to depression seem to have an oversensitivity to negative emotion. So they may pay more attention to negative things. They have difficulty disengaging from something negative. So once they've discovered someone in the corner who's scowling at them, they can't let that go, or they've had moments of doubt, they can't let those thoughts go. They interpret things in negative ways when they could be either positive or negative, and they show biases in memory. So all of their negative memories come very easily to the surface, and their positive memories are sort of hidden away and much harder for them to access. So when something happens, it's easy that's bad. It's very easy to see how it's very similar to all the other bad things that have happened in your life, because those are very accessible. And then there's that oversensitivity paired with difficulty in controlling that information. So you see rumination happening, for example. So inability to banish the negative thoughts and move on and think about something else. So again, it's that trade-off between an oversensitivity to the negative emotional cues coming in, with this failure to regulate and control those emotions when they cease to be useful.
My name is Michael Tooley, and we're now in the psychophysiology recording room. So we have some psychophysiology recording equipment there, which is attached to a bunch of electrodes, which we attach to various parts of the person's face and over various muscles that we're trying to measure reactions in. And then we also have a computer that's presenting an experiment.
All right, so this is where you would see any images that you might be responding to, or
Right.
whatever the task is, really.
Yes. So in my task, they're viewing images, and they're either positive, negative, or neutral images.
So is it a smiley face, or what other sort of images, or something scarier than that?
No, they're a bit scarier than that. I'm looking at very sort of basic physiological sort of responses, and so I guess you think of things, emotional things that are quite salient in maybe an evolutionary old way. And so I tend to use for negative images things like mutilated bodies, attack scenes, things like that, and the pleasant images are actually typically erotica. So with relation to frontal asymmetry and vulnerability to depression, the study I did last was using the startle reflex. So what we did, we presented this very brief, loud noise, which is a white noise. What that does is it induces a startle reflex, and part of that startle reflex is that people blink, or at least the muscle around people's eyes that make their eyelids blink activates. And so I can place electrodes over that muscle, and I can record activity in that muscle. And what you see is that people have a larger blink when they're processing negative information, and a smaller blink when they're processing positive information. The idea is that that when you're processing negative information, you're essentially primed to blink quicker. So it's the startle reflex. There's a number of processes involved in the startle reflex, and it's protective. So the idea being that if you're in a negative situation and you're startled, you blink to protect your eyes. So what I did was present these emotional images, and then I could present startle probes either during the image, and so I'm looking at people's reactivity to the image, and then by presenting startle probes after the image, I'm then be able to look at how long it takes people to come back down to a baseline level of responding. So I'm looking at emotion regulation. What we found there was that people with this rightward asymmetry, so more vulnerability to depression, were actually slower to regulate their emotions, so they took longer to come back to a baseline level.
Which kind of makes intuitive sense, that is exactly what you'd expect to happen.
Yes, exactly. Also, I've followed these people up across a course of a few months and asked them about life stress that they've experienced in the last few months since the experimental session, and also I've measured depressive symptoms. And what we find is that those people that are not very good at regulating their emotions, or that take longer to regulate their emotions in relation to the pictures, actually, when they experience more life stress, will show higher depressive symptoms, whereas the people who are good at regulating their emotions at that first time, when they experience life stress, then they have no change in their depressive symptoms.
It's interesting when you said that was intuitive and that's what you would think would happen. But in fact, this is not something that we've known until this experiment. So what we've known very generally about what that pattern of frontal brain asymmetry represents is largely based on people's sort of questionnaire measures of how people consider themselves to react to emotional information. But again, those sorts of things are very susceptible to people's own interpretation of their own behavior. And so frontal asymmetry, there could be hundreds of different mechanisms that allow it to make somebody vulnerable to depression. And so we've sort of targeted this specific hypothesis that it has something to do with controlling this relationship between information coming in and controlling it and regulating it back down again. And so the experiment is actually really consistent with that hypothesis, but it's actually one of the first pieces of evidence that we've actually had to tie those two aspects of neurological function together.
I'm Josh Foster, and I'm a master's student in the School of Psychology. So you've heard a bit about frontal asymmetry as a marker for vulnerability to depression and cognitive theories of vulnerability to depression. One of the predictions of cognitive theories is that people that are vulnerable to depression might show attentional biases to negative information. So people who are vulnerable might dwell on negative information, and that may be why they go on to develop depression. So in an experiment that I ran recently, I was trying to get at this idea. So people come into the lab and, as you've heard, the frontal asymmetry was measured using EEG. And then they did this task.